So let's go ahead and get started. To shamelessly plug myself, and particularly my students, I needed some good news, and this was literally in my inbox this morning, that our manuscript was just accepted. So very excited about that. Second thing is that on the 17th of February, so that's a couple of weeks from now, on a Friday, the dean of the department, or sorry, the College of Liberal Arts and Sciences will be joining us. And I'll let you try and figure out who she is. Probably the one who doesn't have a clicker. I won't be pointing her out to you. The other thing, of course, is what's happening next Wednesday, our first midterm. What that means is we will have a review lecture on Monday. I think I'll still have some clicker questions there too, at least at the very beginning thereof. But there's not going to be any new material on Monday. All the material that will be on the exam will be what we covered today. So I won't be doing any more new stuff, and that's partly to give me a chance to go back and look at my notes and go, oh my God, I did a horrible job of explaining that the first time around, or something like that. It's sort of a way that I can save myself, as it were, in that whole process. Are there any questions at this point about the exam? I've posted old exams with and without keys. They should be available. Yes? What kind of Scantron do you need? The big blue ones. So those are the ones you need, not the little skinny ones. And the main reason for that is we got a new scanner in the office, and it doesn't take the little ones anymore. They're also way easier to scan so that I have copies of all of your exams. Exactly that one. Thank you. The blue one. Great. Any other questions before we get started? Okay, so again, today we'll basically start off where we left off last time, which was in sliding clamps. These are the processivity factors that are absolutely required. Oh, yeah, sure. Sorry. Pardon? Can you use note cards for the exam? No. I'm afraid not. Plus, how you would shrink everything into a note card, you'd like a huge microscope or something probably to see them. I'll take your word for it. So sliding clamps. These are what the polymerase needs to make sure that it stays on the DNA. And as it turns out, these sliding clamps are also really good at bringing other proteins and other protein complexes to the replication fork where they're needed. So it's not just a process that is interacting with the DNA polymerase. It's also helping bring together a lot of other proteins. One of my colleagues who works on these things thinks of kind of like a tool belt, like the replication tool belt. So it's got the DNA polymerase hanging off here, primase hanging off there, RNase H hanging off on the other side. So interacting with lots and lots of different proteins. Turns out that this particular structure, the ring around the DNA, is extremely similar in all kinds of different organisms. This is the one from E. coli. It's called the beta sliding clamp. There's also PCNA, again, more alphabet soup. Proliferating cell nuclear antigen is what PCNA stands for, proliferating cell nuclear antigen. The reason for that is that when you're proliferating, what are you doing? You're replicating your DNA. And as we'll see, you actually need many, many copies of this. So it's something that's present in the nucleus. You have many copies of it. That's the antigen the antibodies are going to be reacting with that interacts with it. And even viruses have very similar structures. This is the one from bacteriophage T4. And as was mentioned last time, I think, how do you get this ring around the DNA? You have to have a specific other protein complex to do this. The, again, incredibly creatively named clamp loader complex. So this is an ATPase, which will bind to one of these sliding clamps. Again, it's a ring, but at that point, it's not around the DNA. It opens up that ring structure, allows it to associate with the DNA, and then that ring structure closes along with the hydrolysis of ATP. And so that's what you need these clamp loaders for, is they're ATPases. They basically hold that clamp apart till it gets to the DNA, and then let it associate. And once it's on the DNA, it actually stays on the DNA for quite a long time. And how it comes off is an interesting question that no one really has a good answer to. So to risk belaboring the point here, this is a really nice image, I think, of the sliding clamp. In this case, it's the beta sliding clamp. It's only got 2 subunits. PCNA, as I forgot to mention, is actually 3 subunits, which is kind of interesting, even though they have almost exactly the same structure. One's in 3 and one's in 2. And fits really nicely around the DNA here in this space-fitting form, sticking basically into the major groove. And this really kind of rotates around the DNA in the major groove and holding it there. You had a question in the back. Yeah? This is for replication. So this is, we haven't gotten to transcription yet. That's like after the midterms. So don't even bother thinking about it for now, except for the central dogma. No, but yeah, this is just for replication. It's just for interacting with the DNA polymerase. And to risk getting a little overboard here, it's a DNA-dependent DNA polymerase, which means that it binds to DNA, uses DNA as a template, and makes DNA. When we talk about transcription later, you'll see the differences that come out of that. And so, again, what this sliding clamp does is it gets loaded onto the DNA by the clamp loader, interacts with the DNA polymerase, and these DNA polymerases are not processive. They would love to just fall off the DNA and float around in the nucleus if we're a eukaryote or just in the bacterial cytoplasm. But since they're bound to the sliding clamp, they stay there, and they'll keep replicating until down here at the bottom, where the polymerase now hits one of these primers, and then the polymerase falls off. The sliding clamp stays on. And this is also where you get back to that tool belt approach. What do you need now to do once you hit one of these primers? It's RNA. What has to happen? You need to get rid of it. It needs to become DNA. And so you need RNase H, or in the case of E. coli, a DNA polymerase 1 that will get rid of this. And as we'll see in eukaryotes, there's also something called the flap endonuclease, which will get rid of that. Yeah?
So is this pushing the DNA polymerase along?
Pushing the DNA polymerase along. No, it's not pushing the DNA polymerase along. Actually, the DNA polymerase is translocating, i.e. moving along the DNA, due to that hydrolysis that's happening. And so the DNA polymerase will actually bind to where there's a free 3' OH, and that keeps moving as soon as it keeps adding one. So it's actually the DNA polymerase just kind of pulling the ring along in this case. Okay, other questions about this? Good, no, when he says that, what does he mean? There'll be a clicker question, exactly. So sliding clamp seems to be necessary for genome replication because DNA polymerases are non-processive, DNA polymerases have low fidelity, cannot associate with DNA without a clamp, hydrolyzes ATP, and it acts as a helicase. Oh, helicase. I hate autocorrect. That should be a helicase here. Helicase. Ten, 5, clutch. We seem to be pretty evenly divided between A and C. And I'm sorry about this overlap here. So it seems to be necessary for genome replication because DNA polymerases are not processive, low fidelity, cannot associate without a clamp, hydrolyzes ATP, and acts as a helicase, again, typo. No one likes the last two. The question is between the first three, basically. And the question here really has to do with what's meant by processive and what's meant by fidelity. What does processive mean? that it stays on and keeps adding nucleotides, right? Fidelity is what? Makes mistakes. Does the sliding clamp have anything to do with making mistakes? No. Absolutely none. Okay, so B's out. So we're down to binding to DNA or falling off of the DNA. So which one is it? It's falling off, so it's A. Question in the back, yes?
So if a nucleotide is added directly with the sliding forward and act as a kind of like a checker.
So I'm not sure I quite follow your question. Sorry.
If you had a mismatched base pair,
yes.
as you're adding nucleotides to it, the plant is so specialized and small enough, would it pass over a mismatched base pair or would it get stuck until that base pair?
Okay, yeah, so the question is, as the clamp is sliding along, would it basically recognize a structural problem that has happened in the DNA? Turns out that for mismatches, no, but when we talk about DNA damage after the midterm, yes. So some of both. And it has a lot to do with, again, the structure of the DNA. It's not so much the sequence, it's the actual structure. And clearly, mismatched base pairs are going to have a different structure. So it's that structure which is being detected. Yeah, sure.
Can DNA polymerase associate with DNA without the clamp?
Yes, so DNA polymerase can associate very well without the clamp, and those were the original experiments that they did with E. coli, just purifying those individual polymerases, and they showed that they needed a template, they needed a primer, and nucleotide triphosphates, but that was it. You didn't need anything else. Now, to make sure it's processive and actually replicates the genome, then you have to have these sliding clamps.
So would you see it without the clamp in a smaller organism?
So, question here is, would you see it without a clamp in a smaller organism? Okay, so I'm going to paraphrase a little bit here, pardon me. But the smaller organisms, the organismal genomes that we've talked about, even the smallest ones are a couple of 100,000. So it turns out, yes, you always do have a sliding clamp in those. We want to think viruses as organisms, which of course are the coolest things ever. Then in some cases it seems you don't, particularly for the ones with the very small genomes. But in some cases you do, and I showed that bacteriophage T4 sliding clamp, which was actually one of the first ones that people studied. That's a viral genome, but it's relatively large. It's a couple of 100,000 base pairs in length, so it probably has to do with that. Although in most cases the viruses are in cells that already have sliding clamps, they just use the cellular one. So, but as far as I know, there are no organisms that do not have a sliding clamp that aren't dependent on some other part of the other machinery. But it's a good question, and it turns out that the way people find these things, we'll get to in just a second when we talk about mismatch repair, has to do with the mutations and how you look at those. So what's present at a replication fork? We basically talked about all these things, but I wanted to emphasize what these things really look like because it's a little confusing, and wrapping your brain around it, at least for me, was rather challenging to start with. So I want to spend a little while going over this. We've got our leading strand here. We've got our lagging strand over here. Leading strand is the one that's just going from 5' to 3'. You've got a polymerase. You've got a sliding clamp. This can just keep going and going and going as long as you have a single-stranded piece. How do you get the single-stranded piece? The activity of the DNA helicase, which is now wrapped around one strand instead of the two strands, but again, this loop structure. So that's pretty straightforward. On the other hand, you've got this lagging strand, which is being replicated semi-discontinuously, also Okazaki fragments. What that really means is you've got to start doing replication many, many times on this lagging strand. And it turns out this is exactly what happens also at the origins of replication, that hopefully we'll get to at the end of the lecture today. You have a DNA primase, which again is an RNA polymerase, putting down these little blue RNA bits. Then you have a clamp loader, which is this green piece right here, together with a sliding clamp that's open. So as soon as you get this primer put down, now the sliding clamp can get associated with it. You can get a DNA polymerase that will now extend. Now, all DNA polymerases are going from 5' to 3'. None of them are going back the other way because of proofreading reasons. So how do you put all of this together in one complex? Basically, what this is they call it sort of a trombone model. And we'll look at this. We've got a great animation that we'll take a quick look at in just a second here. But basically what happens is once this primer is formed, then you can have a DNA polymerase that will go along it until it hits the next primer. Then this DNA polymerase will actually fall off, wait for the next clamp to be put down, and then extend from there. That also means you need a lot of the single-stranded binding protein, because all of the single-stranded binding protein is basically waiting for this polymerase right here to get to the end of that Okazaki fragment and then bounce back and take a look at it. Yeah?
Is it the same polymerase molecule, or is there just a lot of polymerases floating around waiting to associate?
So the question is it just one polymerase molecule that's hanging out right there at the replication fork, or are there lots of DNA polymerase molecules floating around and then come in and associate? Turns out they're all together, and they're all stuck together. And people showed this really nicely early on in the process. You remember we talked about DNA polymerase 1 that Arthur Kornberg found? Well, the reason he found it was because it was the one that was the most common in the cell. Turns out it's DNA polymerase 3 in E. coli which does this. And that DNA polymerase 3 is at very low concentrations because you just have one leading strand, one lagging strand polymerase, but always associated with each other at the replication fork. And that actually is shown here in the blob up at the top here, but people have looked at leading strand and lagging strand. This is an electron micrograph where you have leading strand and lagging strand, and then this loop structure out here, and this is now your replication fork complex. Slight cartoon here. Again, polymerase, DNA polymerase 3, DNA polymerase 3, all connected together with these protein-protein interactions. And again, this is something we're going to talk about again and again and again through the course. Protein-protein interactions with a clamp loader, and then a sliding clamp. And if you just purify these enzymes, these enzymatic activities, you'll find all of those things associated with each other. And so another way of looking at this is here. It's basically exactly what we just looked at, only mirror image, looking at it in the opposite orientation. This one's a little bit closer to what the real structure actually looks like. Again, primases, helicases, sliding clamps, and leading strand polymerase, lagging strand polymerase. Here it's got these single-stranded binding proteins bound here, should be bound over here on this side as well. What happens with a structure like this, this is an image from a paper, if you want to take a quick look at it, should be freely available, is when you have your lagging strand polymerase here gets to the primer for the next piece, this one at least has the single-stranded binding protein on it, it then will dissociate from the sliding clamp. You see it still stays attached right here. So that was your question about coming in from somewhere else. And now we'll move to the next sliding clamp, which what the clamp loader just put on the new primer, and extend from there. So what does this look like? This is one of my favorite videos for looking at, oh yeah, let's get rid of this wonderful post here. And take a look at these molecular visualizations of DNA. This first part is that part where we have nucleosome condensation. If you go a little bit further, now we'll look at the DNA replication process. Load up the full screen. So here is our complex. Right here, there's the lagging strand like this, your leading strand is down here. There's your helicase pulling the strands apart. There's that clamp loader coming in from the back, putting down a sliding clamp. The purple is the polymerase. Right here, lagging strand, so it's bouncing back and forth in this trombone, which is the template for that lagging strand. Spooling out, so you get to the end, go and start again. Process again and again and again. Again, this is, I think it's worth taking a look at this if you're wondering how the process works. It's missing a couple of things. Most obvious is likely single-stranded DNA binding proteins. And then there are a few other things that we'll talk about in just a second. Yeah.
Is the exonuclease that's part of DNA polymerase, is that working as the DNA is being replicated, or does it go back over it once it's finished?
Okay, so basically what you're asking is, when is proofreading happening? When is that backing up exonuclease going on? So again, we're replicating from 5 to 3. What about the 3 to 5? Going back in the opposite direction. That's happening while this is moving forward. And so let's put in the wrong nucleotide, take one step back and keep going. Yeah?
Is helicase always on the lagging strand?
Pardon?
Is helicase always connected to the lagging strand?
So the helicase, it seems to be sort of right in the middle, but whether it's actually wrapped around the leading strand or the lagging strand, you have to think about it's going to be the template then for the leading strand or the template for the lagging strand, just pulling those two strands apart. To my knowledge, it's always a template for the lagging strand, but that I'm not completely sure about. Ask Dr. Purcell, he would know. Yeah.
Are the sliding clamps used again after? Once they get to the fragment?
Okay, so the question is basically, are those sliding clamps used again once they get to the end of the Okazaki fragment? For DNA polymerase, no, because it's back doing its next Okazaki fragment. But in terms of getting all of the enzymes that you need to get rid of that RNA primer, yes, it's still being used. And it may actually be the presence of one of these sliding clamps may also be a marker for the lagging strand, as you were asking about the back there. It's the fact that you have one of these sliding clamps associated with it, that means that that's a lagging strand versus being a leading strand. And that does turn out to be important for a couple of things. Okay, so let's continue. Then I highly recommend taking a look at that video a couple of times, at the very least, to go through and take a look at it. So that was a very general sense. Unfortunately, we're talking about eukaryotes and bacteria, to some extent archaea as well. They've all got different names for things, which is really ridiculous, but this is what is happening. So we already talked about PCNA versus the sliding clamp. Of course, a nuclear antigen, so it can't be bacteria. Bacteria don't have nuclei. Then we have a clamp loader, which is the RFC complex. Not DNA polymerase 3, because again, we're not talking E. coli. This is not DNA polymerase delta. I think there are up to about 8 or 9 different DNA polymerases that you find in eukaryotic cells. But DNA polymerase delta and also DNA polymerase epsilon are the main ones which are involved in the replicative polymerases that you have in eukaryotic cells. RPA is the single-stranded DNA binding protein. We have a helicase. In this textbook, it called it T antigen, which is an interesting aside. T antigen is actually a tumor antigen from a viral infection, SD40, which is one of the first studied. It's actually now MCM, or mini chromosome maintenance protein, also named something called GINs that we won't talk about for the rest of the classes and movements. But for a long time, people studied replication by looking at viruses in terms of studying replication. So that's where that comes from. It's a helicase, basically does exactly the same thing, but as you'll notice here, up in the back, now we've got the helicase wrapped around the template for the leading strand rather than around for the lagging strand. So, and I can't remember exactly the detail on how all of that is getting put together. So, on the lagging strand, there are a couple of things that are a little bit different. We've got our DNA primase, which is putting down your RNA primer. But in eukaryotes, you have an extra DNA polymerase. This is DNA polymerase alpha. And basically, all the DNA polymerase alpha seems to do is extend primers a little bit more after the DNA primase. So this is something, again, that doesn't exist in bacterial replication. It's just present in eukaryotic replication. Then, at the end here, when you end up at the end of your Okazaki fragment, you're trying to get rid of your primers, there's RNase H, just like you have in E. coli. But there's also another new thing, which is called FEN1, down here at the bottom. Now, FEN1, what does FEN stand for? It's the flap endonuclease. Right? Talk about exonucleases, they chew in at the end of DNA, right? Well, an endonuclease cuts in the middle of DNA. So, okay, an endonuclease cuts in the middle of your DNA. But what's it cutting into? It's cutting a flap. What the heck is a flap? That flap is this RNA, which actually gets moved a little bit out of the way by the DNA polymerase, which is coming through. And so a little RNA flap, this extra piece, that gets cut off. And so it's a flap endonuclease that is basically doing that extra job. There's also this topoisomerase out here at the end. We'll talk about those in just a second. So I find it's useful.
They said the flap was just some extra pieces of RNA that gets cut off.
It's the RNA that's left from the primer that gets cut off. It basically seems to be kind of a redundant system. You've got RNase H that cleaves RNA within an RNA-DNA hybrid, and the flap endonuclease is going to cut that off. Now, last time we talked about the really bizarre aspect of DNA polymerase 1, that as well as having a 3' to 5' exonuclease, also has a 5' to 3' exonuclease. So it chews up nucleotides that are in front of it. So it can also get rid of the RNA primers. But that's only present in bacteria, it's not present in yeast. Yeah, back. Yeah. The question is, does DNA polymerase alpha extend the primer with DNA or with RNA? It extends it with DNA. So it is in fact a true DNA polymerase, unlike DNA primase, which is RNA. Okay, so again, I find this, I'm not going to go over this in much detail, but I find this a really useful reference to try and remember all of the different funky names. Again, the alphabet soup that comes with all of these things. DNA polymerases, 3 versus delta, primase. Anything that's in red here is going to be different between the two. All alpha, you don't have the equivalent. In bacteria, DNA polymerase 1, you don't have the equivalent in eukaryotic systems. Fen1, you don't have the equivalent in bacterial systems. So, useful to put these things together. Any more questions? Good? No? Turn this back on. Which of the following proteins binds to single-stranded DNA? RPA, RC, PCNA, Fen1, RNase H.
No.
Classic kind of question that Stedman would ask on an exam. Not that any of those would happen in your future.
I would say RNase because that's getting rid of the RNA fiber before the. That actually, so that would be the double stranded. Oh, I guess Fen1 because if it's cooking off just the RNA. I'm just going to put that in.
But it's two single-stranded DNA and one of those RNA.
RPA, again, these are the kinds of things that I would expect you to remember. Again, I'll give you that table on that last slide. SSB, RPA. What's RC? The clam loader. PCNA is the clam. Fen1 is flat endonuclease. RNase H is RNase H that cuts the DNA RNA hybrid. Yes?
So we should know the acronyms as well as the full names?
I would know the acronyms as well. It's also very confusing. One of the things I didn't talk about is what all these acronyms stand for. RPA, RFC, etc. If you really want to know, it's replication protein A. Like, that's useful. And replication factor C. Again, totally not useful. I find that PCNA is somewhat useful because there's a whole bunch of it. And thinking about it from that point of view, and then actually makes sense flat endonuclease. So I'll try and the ones that I think make some kind of sense, I'll talk about. Those that don't, we'll just use them. Yeah.
Yeah.
okay, so the question here is actually a very good question having to do with terminology. Is RPA a single-stranded binding protein? Yes, it is a single-stranded binding protein. But there's also SSB in E. coli, and they would just call it SSB. So single-stranded binding protein, SSB in E. coli. There's another name for it, we're not going to get into it. It's different from different bacteria. But yeah, so but if you talk about SSB, that would be E. coli. RPA is going to be for eukaryotic. Yes?
So is that the same structure or same function with a different structure?
So yeah, the question is RPA have the same function with a different structure? And the answer is exactly yes. It turns out you might have seen there are actually 3 little pieces that came together in some of those cartoons. So it turns out that the RPA is actually 3 different subunits that come together in terms of making that particular structure. If you remember from last time, SSB is at 2. It does seem to be sort of a convergence. You have something that has to bind to DNA. If you look at the high-resolution structure, then it turns out there are some similarities between the two, but in general, they're pretty different. Yeah?
Are the single-stranded binding proteins recycled as DNA is replicated, or are they reproduced? How does that happen?
So the question is what basically, sorry, I'm going to paraphrase here, but what happens to single-stranded binding protein after you have replication? So you can't have those single-stranded binding proteins there and still replicate, put the DNA on there. So it turns out the polymerase actually bumps them off. But they're still kept, they're still around, and so they can bind back on as soon as the helicase opens up and makes some more single-stranded DNA bonds. Yeah.
I thought the text said that it doesn't cover the base pairs, though, or the bases, the single-stranded binding protein.
The single-stranded binding protein doesn't actually cover the bases, so you're exactly right. But in order to have the polymerase coming along and the sliding clamp, it's not going to be available to be able to do that. Yeah, so it's not actually binding the bases per se. If it were binding bases per se, it probably would be more likely to be a base-specific binding, and you don't want that. You want something which is very general. So, yeah, but you do need to have it displaced in order to get the polymerization to happen. Okay, I need to select the correct answer here, otherwise all these people who got it right won't get points for it. Okay, so again, some of you may have noticed at the beginning that, when I'm looking at the beginning, when we're looking at these replication forks, there was this funky thing off to the right, at least if the fork was on the left, called topoisomerase. What the heck is a topoisomerase? Well, it's an ACE, that means it's probably an enzyme, but what's it doing? So it's changing the topology. I don't think that topology in math. I did not, so that makes at least 131 of us. So what topoisomerases do is they change the topology of DNA. Now, why do you need to change the topology of DNA? Well, what happens when you replicate and you make single strands, and particularly with helicases, you pull these 2 strands apart. If you're pulling the 2 strands apart, you're untwisting them because they're wrapped around each other. Well, if you're pulling them apart and the other end is stopped, what happens is that has to compensate for this untwisting. So you're untwisting this way, you're going to have to twist again in the opposite orientation. And we can look at that here. Here's just a case of, we've got our untwisted. If I twist it a little bit, which is just turning around here, that will then wrap together. And that's your superquadron that's happening. So we have extra things, extra wraps which are happening due to the unwinding process here. Now, if you don't have a topoisomerase that can change this overwrapping, then what happens is the polymerase actually gets stuck. Let us the helicase, because there's too many of these things. It's been so overwound you can't wind it anymore, and you get things stuck. And so what you need are topoisomerases to change the topology of your DNA. And so that's the reason you have these topoisomerases, to take care of that. It's just the one from our textbook with basically exactly the same thing. There are 2 flavors of topoisomerase. Again, incredibly creatively named Topo 1 and Topo 2, but there's actually good reasons for that. Topoisomerase 1s cut 1 strand of DNA, and topoisomerase 2s cut 2 strands of DNA. And what that looks like is here. There are two possibilities for dealing with this kind of, overwrapping, underwrapping. So you can just change one strand relative to the other. This is what happens with topoisomerase 1. Because you have a double-stranded DNA, which gets cut through this type 1 DNA topoisomerase that uses an OH on a tyrosine side chain. It's a very typical kind of reaction. That OH now is basically pretending to be the 3' OH that's present in your DNA backbone. So it binds here. Now you have a 3' OH, and the DNA just wraps around itself. This is a reversible reaction, and so the OH is exchanged. Now the topoisomerase is no longer bound to the DNA, and the DNA is reattached here. So what you're doing is you're cutting one of these strands, wrapping the other one around, and putting that strand back together. So that's one way to deal with this overwrapping process. There's another way, which is topoisomerase 2. And so what topoisomerase 2s do is they take double-stranded DNA, cut both strands, pass another double-stranded DNA in between, and then put those strands back together. And again, mathematicians will tell us that it's actually absolutely equivalent topologically, and you can change these things back and forth. We're not going to talk too much more about linking numbers. And look at old exams, and we'll say linking numbers and that, twist and ride. We're not going to get into that and get people too confused. But these are equivalent. That's all we need to know. Is it passing double strands through double strands, or taking one strand and wrapping it around, are equivalent to each other. So these are the ways that you can deal with that extra winding that you get just because you're pulling the two strands apart. So you have to have topoisomerases, otherwise you can't continue to replicate. Yeah.
Is there a preferential way in which these are used by the replication machinery? Or?
So the question is here, basically, when you have a replication fork, you have a topo 1 or a topo 2 that's associated with it. And the answer is, you actually have both. Some of them have topo 1, some of them have topo 2. And it seems to vary depending on different replication forks. We'll see an example. We actually have to have a topo 2 in about 12 minutes. Assuming that I get there. So we've got a replication fork. We've got leading strand, lagging strand, etc. And the proofreading of all the polymerases. The polymerase has its nice checking the structure, but occasionally the wrong base gets put in. And we talked last time about the fidelity that you get from just the DNA polymerase. And the fidelity you get from just the DNA polymerase is about 1 in 10 to 1 in 100 million. Well, what's the general mutation rate? 1 in about 1 billion. So how do you get from one to the other? Turns out that there's this wonderful repair mechanism, which actually happens right after replication, which is why it's included now rather than when we talk about DNA repair a little later on, called mismatch repair. And basically what this is it's another quality control system which says, hey, the wrong base got put in here, we need to take it out and put back in the correct base. Now usually these are actually normal bases, so C's, A's, T's, and G's, they just get put in incorrectly, by mistake somehow. And quite often that's because of the tautomerization that happens with the individual nucleotides. Again, amino versus amino groups which are there and are going to base pair slightly differently. So how do we know about these things? Well, the first thing was that people looked at E. coli that didn't replicate well, didn't make 1 in 1 billion mistakes, made 1 in a couple of million mistakes. And you can see that by looking at the various different mutant phenotypes of the organism. And so they said, okay, well, what's wrong with this particular one? Because, again, geneticists love to break things and figure out what they've broken. They just named all of these mutants based on the phenotype that they saw. And we'll talk much more about this as we move on through the course. Again, it's a way of trying to, at least for me, wrap my head around all these strange names. So they called all of these different mutants were mutants. So if you have a change in this gene, it causes a higher mutation rate. So what we end up with, mut S, mut L, lots of different genes, and mut H, which are involved in this extra quality control process. We now know how they work, and that's down here at the bottom. There's some kind of mistake that happens. This gets recognized by the mut S protein. Here's the mut S protein, actually a high-resolution X-ray crystal structure of it, bound to DNA. What's it recognizing? It's recognizing the structure of the wrong base. It recognizes the structure of the wrong base, then, together with the mut L protein, it will cut out a piece of DNA down here that will then get remade and hopefully not put in the wrong base when this happens. So this is great, it's wonderful, but there's a big problem. There's a mismatch in your double-stranded piece of DNA. How do you know which is the wrong one? They're normal nucleotides. So it turns out there are two generally different ways of dealing with this. E. coli is smart. Probably has had more years of evolution to deal with this. What E. coli does is, after its DNA has already been replicated, it puts methyl groups on. So here, the GATC, starts a little hard to see behind the clicker thing here. GATC, all of the GATCs get methylated, adenine, after you've done replication. But this happens well after you've done replication. So what that means is any strand that has a methyl group on it, that's an old one. That's the right information. You want to get rid of the new strand where you put in the wrong nucleotide. So that's how the E. coli mismatch repair system figures this out. This methylation versus one. On the other hand, eukaryotes don't do this. They methylate their DNA, but for all kinds of different reasons. Also to do with silencing, again, we'll talk about much later on. So what the eukaryotic systems seem to do is they look for nicks. Now, what's a nick? A nick is a space in your DNA where there's no covalent bond between the 3' OH and the 5' phosphate. This is what you see after you've gotten rid of those primers, the RNase H and flat endonuclease. You cut, the polymerases come along, the polymerase is associated, the DNA ligase to put those together. Before that DNA ligase comes along, then you have a space that's still in the DNA that can be detected, and certainly now for the lagging strand, it's really obvious that that's where you have a problem. Leading strand is a little harder to understand exactly how the replication machinery knows that one of them is the original strand, the other one is not. Turns out that semi-discontinuous is not just the lagging strand, the leading strand is also somewhat semi-discontinuous as well. And so there are also nicks in the new piece of DNA that's made on the leading strand as well. So I've got nicks down here at the bottom, mut H, eukaryotes, HNPCC, what the heck does that all mean? So mut H, again, these are names that have come from E. coli mutants. These are things that undergo very high mutation frequencies if this gene is mutated. Mut H is actually the thing that comes in here in this strand removal step and gets rid of this extra piece of DNA. In the eukaryotes, those nicks just seem to be there because, again, of the lagging strand synthesis. HNPCC, what the heck is HNPCC? Hereditary non-polyopsis colon cancer. Turns out that in this particular kind of colon cancer, which is one of the more common kinds of hereditary cancers, there's a mutation in one of the genes that's involved in this repair process. So just the fact you are missing this gene means you're going to have much higher rates of cancer. because your polymerase is not putting in the nucleotide appropriately. It's not correcting the ones that you say. It's not correcting the ones that it's putting in correctly. So these are very directly connected to each other. Yeah.
So make sure I got this. The NICs indicate the new strand. So the new strand is what's going to be removed, and then the old strand remains the template.
Yes. So
that indicates that that's the one that needs to be removed in the mismatch.
Yeah. So I'll just repeat this, but you're exactly right. And so the new strand, the one that's just been put in, where you put in the mistake, because that's what you're trying to fix, that's the one that has the nicks in it. That's the one that gets taken away and resynthesized. It's easy, quote unquote, to be E. coli. This is a strand that's not methylated. If you're a eukaryotic cell, you've got to see where those extra nicks are. Yeah.
Are those methylated restriction sites?
The question here is, are those methylated restriction sites? That's way ahead of what we're going to get to right now. It turns out, yes, they often are restriction in nucleosides, and that's what we're protecting your DNA or other DNA. But we'll talk about that when we talk about chapter. Yeah.
How much is actually cut out? Basically, or does it matter?
So the question is here is how many bases are actually removed in that process? Turns out that varies a lot. Probably less are cut out in terms of E. coli. It's got this nice mechanism that clearly which strand you want versus the other. Whereas if you're talking about Okazaki fragments, that can be much more challenging. Turns out Okazaki fragments in eukaryotic cells are usually a lot shorter than the Okazaki fragments you have in bacteria, probably because of that to some extent. And those nicks which are there just seem to serve as a way of being able to do this mismatch repair process. Yeah?
In the upper right-hand picture of the second half, there's a nick with a little triangle indicating where the mismatch pair is. So when the mismatch pair occurs, there's no hydrolysis between the 3' and the 5' on either side of that mismatch pair. They're not actually, the backbone of the DNA is not connected.
I'm not sure I completely follow your question, but you do actually need to, in this step here, just to correct me if I'm not answering your question. This here is just a bumped out piece, because the structure's wrong because you don't have appropriate base pairs.
But the backbone is still
The backbone is completely normal at that point. So you have to have another nick that takes place, and in fact, because the bacteria, there are two nicks that form. There's one here, and then one on the opposite side of this one. And that's what this mut protein does. It does that nicking in order to give you, it'll give you a cut on this side and a cut on that side of that red strand, which is the one we're going to get rid of. You also need a helicase to get rid of that, and then it gets resynthesized. Yeah, but in your normal mismatch, you're going to have a normal backbone because the polymerase has just gone over that. You already got all your 5' to 3' linkers. Okay, quick review for all of these things. Which of the following hydrolyzes ATP? Topo 1, Topo 2, Mut-S, Mut-L, Mut-H.
Yeah, used to pull the two strands apart when that used to have hydrolysis. Right. Right, yeah, looking
Okay, Topo 1 versus Topo 2, Mut-S, Mut-L, Mut-H. Okay, so no one likes the mutes, which is good because they're associating with the mismatch. They're finding the right place. In some cases, they're actually cleaving the backbone. That's a hydrolysis reaction. But if you think about it from an entropic point of view, is hydrolysis going to be energetically good or bad? Actually, pretty good. Hydrolysis is pretty easy to do. Certainly from a chemical point of view. It's that polymerization, which is really hard. It's forming the bonds, which is really hard. So, none of those. Okay? Topo 1 versus Topo 2. Topo 1 is actually cutting and religating the DNA, but it's that OH which it provides, puts in, and just an exchange reaction which takes place. You don't need any ATPase for that. So, you do need it for Topo 2, which is literally pulling those two strands apart and putting them back together. So that's what you need ATP for. Okay, I want to finish up talking about initiation. This is where to start. There are lots of things that have to happen. You've got to put together that whole replication fork machinery. You may need their leading strand polymerase, your lagging strand polymerase, your clamp loader, etc. But the main thing you need to do is get those two strands apart. Because if you don't have the two strands apart, your primase can't do anything, nothing else is a possibility. So, what you need to do is hold those two strands apart. It really is that helicase, getting the helicase onto the DNA and pulling two strands apart. Once you've done that, you're going to get replication taking place at that particular position. In eukaryotes, there are literally hundreds of these things, in fact, some cases thousands of them, but basically hundreds of nucleotides that are usually pretty rich in AT base pairs. Why rich in AT base pairs? Hydrogen bonds. It's actually easier to pull apart relative to each other. But to be able to figure out where these are, you need the so-called initiator proteins. We'll talk about in more detail a little bit later on. We move into the individual ones. And then, once again, you have this two strands pulled apart, your primer being put down. Then you can go to your elongating state, which is where your leading strand is leading down here. Lagging strands will start a little bit later. And you have processive replication. So what are these initiators? Basically, all that it is a DNA binding protein that's going to bind to, protein complex in some cases, it's going to bind to an origin and say, hey, this is the place where you want to start. And once it's done that, again, through protein-protein interactions, how many times do you say protein-protein interactions in this course? We're probably hundreds. Brings in the other proteins which are necessary for pulling those two strands apart, the helicases, and then bringing in all of the other proteins which you need to get replication to start. Very well known in E. coli and in yeast. So let's take a quick look at E. coli, the origin of replication, also known as ORIC. It's the replication start on the chromosome, that's where the C comes from, and it's literally where you start replication in E. coli. E. coli has one origin of replication. One and only one origin of replication. And then, some of you may remember, it's about 4.5 million base pairs. So, takes off there, you have replication initiates. The two replication forks go all the way around the genome, it's a circular genome, and gets to the terminator at the opposite end of the genome. And these two replicated genomes have to somehow come apart. To have the two replicated genomes come apart, this is where you need a topoisomerase type 2. Because what ends up happening is as you replicate, you end up with two circular pieces of DNA. They're actually interlinked with each other, kind of like chain mail. These two linked rings or a chain. You have to undo one and reattach it so that the cell can now divide, and each of them will have a chromosome so they can go off and do their own thing. So you have to have this topoisomerase for separating them, but at the same time, you have 450,000 twists that you've undone in terms of replicating that also need to be taken care of. And this can be done by Topo 1 or by Topo 2. Yeah?
So since this only has one origin replication, does that, if there's a problem when it's initiating and when it's firing, does that mean that replication won't happen for that cell? I mean, does that, will it, if there's a problem, will the cell find a way to find another site to start its one origin replication, or will it fix that one site, or does that just kill replication for that?
Yeah, so the question here basically is if you get rid of one of the origins of replication, can it actually use another one? In many cases, really no. It's very hard for E. coli to do that. On the other hand, eukaryotes are really good at using alternate origins of replication. Yeah.
If this can use both 1 and 2 topoisomerases, what's the point of having both? If they can use either one.
So the question is there, why have 2 kinds of topoisomerases? Topo 1 and Topo 2? The answer is, don't ask why questions in biology. But probably it has to do with the fact that Topo 2 does require ATP, and you do need it for a couple of cases, like here, at the very end of replication. Topo 1 doesn't require ATP, so it's going to be a less energetically demanding one. So that's probably why. But whenever I say why, it's always going to have to be probably. Yeah.
In like longer strands of DNA, is Topo 1 used more than Topo 2 because it's energetically favorable?
Again, the question is it or isn't it? And the answer is, it seems to be both. And getting back to the evolutionary, the why question, as long as it works, it gets used. It doesn't have to necessarily be the optimal solution. Yes?
Does intertwined DNA help stabilize the cell during mitosis? It's like a stopping checkpoint to make sure that organelles are disposed properly.
Okay, so the question is, does this help for mitosis? Yeah. This is E. coli, so it actually is not going through the normal cell cycle that you think about from a eukaryotic cell. But it does turn out that this, these cross-linking between the two is rather important. We'll talk about that later on. Yeah?
That's a tough question. Are there equal concentrations of them, or even more of one and less of the other? Instead of 1 and 2?
Oh, okay, so sorry. At the beginning of that question. The question is, you have different concentrations of Topo 1 and Topo 2. I'm going to hem and haw on that. Partly since it depends on your growth phase, depending on where you are actually growing. From organism to organism, it's different. And there's actually a lot of Topo 1s and a lot of Topo 2s. So it's really hard to separate those two relative to each other. So the answer is, it varies, yeah.
Are there situations where Topo 1 is necessary specifically?
So the situations where Topo 1 is specifically necessary. In theory, you could get away with Topo 2, but again, you need ATP hydrolysis for Topo 2. So probably, and a lot of things that people do is they do mutations, again, like we talked about these mutagenic phenotypes. And so I'm trying to remember what exactly the phenotypes are of some of those Topo 2, but I'll check. Okay, so we need to get to eukaryotic initiation. So we need to move through a couple more of these things. But basically, the process here is just now going through what the proteins are involved in this process. Unfortunately, this is called DNA, B, and C. Again, not very useful. But the initiator, DNA A, this is an E. coli, binds to AT-rich sequences and forms a structure, a DNA protein structure, which now allows this most important protein here, the helicase loader. We had a question up in the back, I think, last time about how do you get that helicase around that template strand? Again, it's a loader. Again, it's an ATP-dependent process. We'll open up the loop form, which is the circular form of your DNA helicase, and clamp it down onto the DNA. Once those two things get pulled apart, you've got DNA primase, etc. Once you get started, you're good to go. So it's really all about this initiator protein and then protein-protein interactions with your clamp loader. DNA A is what happens in E. coli. The, again, incredibly creatively named origin recognition complex in eukaryotes, or ORC. So what do these things actually look like? We've got some really nice structures now of where you have the DNA at the origin bound to different proteins. So ORC that you have in eukaryotic systems, turns out RPA is a very similar one. DNA A, if you look here, these protein structures are actually really pretty different relative to each other, but they're still serving the same role. Finding that origin, protein-protein interactions, interacting with it. Some viral proteins look even more different relative to this, but they still do exactly the same thing. So it's a case of, you need to get those two strands apart, You have to have an initiator protein there, but then once that's bound, you're good to go. Licensing. So, as I mentioned before, and we were talking about this too, in E. coli, you've got one and only one origin of replication. That's because you only have the sequences that DNA can bind to in one place in the genome. That's great. It's wonderful, but it's that one place in the genome. Once you've replicated that particular place, you've got 2 copies of it. What do you do now? Does DNA bind to that again and recruit the nuclease loader, which pulls the 2 strands apart and you start again? That can be a real problem because you've got all these different replication intermediates going on. So how does E. coli again know that it's just used that particular origin of replication? This gets back to exactly the same way that it knows which is the new strand and which is the old strand. After you're done with replication, what do? You methylate. You've methylated, now you have those old strands which are methylated. Once they've separated, you only got what's called a hemimethylated form. That's because one of the strands is methylated, the other strand is not methylated. And so that's how E. coli knows that it doesn't bind. What that says is DNA A is only going to associate with completely methylated DNA. It won't associate with hemimethylated DNA. No DNA association, no helicase loader, no helicase, no replication. So that's bacteria. What happens in eukaryotes? Eukaryotes have a lot of problems, other than the president. Eukaryotic polymerases are slow. The genomes are really big. How do you deal with this? The only way to deal with it is to have lots and lots of origins of replication. And so these are also called autonomous replication sequences in yeast. And basically, they're present all throughout the genome, and they seem to be not terribly sequence specific. And that means you can use a lot of different ones and get away with it. Quite often what you see are a whole bunch of origins of replication quite close to each other, and a big gap, and then another set. The way you can see this is just by looking at DNA that's being newly replicated. You can do this by using radioactive nucleotides and looking at where that DNA is actually being made. And in fact, in that process, you can see that you have lots of different origins of replication. So you do this in E. coli, you have one origin of replication. You do it in a yeast cell, in a eukaryote, you see multiple origins of replication. There are other ways of looking at that we're not going to get into, so we'll skip these 2. If he doesn't talk about them in lecture, they're not going to be on the exam. And just wanted to talk about the last step here, which has to do with the actual initiation of replication at any one of these particular origins of replication. Mentioned before, ORC, the origin recognition complex. This is your initiator. This is where you actually end up starting. So just to finish up right here, you've got double-stranded DNA and origin that gets recognized by the origin recognition complex. Then you have these 2 funky proteins that come in and bind to this origin recognition complex, CDC6 and CDT1. CDC stands for cell division cycle. And if you have a mutation in one of these, you have problems with your cell cycle. So that's where all that naming comes from. So CDC6 and CDT1 are the licensing factors. So they're basically the equivalent of the methylation that you have in your E. coli DNA. Because what happens is these guys get put together. You get your helicase loaded, which is your MPM. Then you have helicase activity starts here, but it's this phosphorylation step that happens right here. Phosphorylation happens to the ORC complex, but also to CDC6. What's doing this phosphorylation? It's cyclin-dependent kinases. So in the cell cycle, how is the cell cycle driven? It's driven through phosphorylation. And that phosphorylation is due to these cyclin-dependent kinases. Cyclin-dependent kinases will only recognize this initiation complex if you have CDC6 and CDT1. And then that puts the helicase together. When you have phosphorylation, phosphorylates the ORC, which says, hey, you can't associate with CDC6 and CDP1 anymore, and CDC6 is phosphorylated, which ends up leading to degradation through ubiquitination, E1, E2, etc. So that's what we'll get to on the midterm. Real quick question before everybody runs away.
Polyubiquitination, does that usually lead to degradation?
So polyubiquitination, yes, does usually lead to degradation, and that's exactly what happens in this case.
We actually talked about the helicase loading. It was like, oh, it's there.